In the last lecture, we looked at the big picture of how genetic information that's stored in DNA flows to proteins via the intermediate messenger molecule, RNA, through a set of processes that were described by what we called the central dogma of molecular biology. In today's lecture, we're going to return to DNA and ask how information about protein structure actually is encoded in DNA in the first place. To remind you one more time, there are two properties necessary for DNA to act as an information-bearing molecule in living systems. It has to be able to be replicated accurately, so copies can be transmitted, and we've looked at that in detail now. And it has to provide a readable code for storing information. Today, we'll begin to look in detail at the nature of that code and how it works. Now, let me give you an example of a simple code. You may or may not be familiar with the cryptoquote puzzle that occurs in the comic sections of many daily newspapers. If you're not familiar with it, it involves some famous or amusing quote that's had all of the letters in it substituted for other letters. For example, all the A's may be substituted with W's, all the B's with P's, and so on. The substitutions are always consistent. That is, W will always be substituted for A, P always means B, and so forth. And the puzzle is solved simply by figuring out the correspondence of letters, making the appropriate back substitutions. This code is a very simple code because it just involves finding the equivalence of single symbols, which are both in the same alphabet. The code we need to direct protein synthesis has to be more complex than that for two different reasons. The first reason our code needs to be more complex is that DNA and proteins are very different kinds of molecules. Even if we could imagine a code that establishes a relationship between the bases in a nucleic acid and the amino acids in a protein, which of course we can, how could this correspondence work on a molecular level? Remember that the trick to DNA replication and to RNA transcription is that there's an inherent correspondence between complementary base pairs, A always pairing with T, C always pairing with G. And this correspondence, you'll recall, is because those base pairs actually have a highly stable, a maximally stable interaction depending on the hydrogen bonding that occurs between them. Now, is there some way to get a molecular mechanism that could achieve a similarly predictable interaction between a particular amino acid and one or more particular nucleic acid bases? The fact that proteins and nucleic acids have quite different biochemical properties in general actually suggests that this sort of direct molecular correspondence, analogous to the complementary base pairing in DNA and RNA, is unlikely to occur. What we need is for some way to have nucleic acids and proteins communicate, maybe through some translator who speaks the language of both. Francis Crick actually was the first to propose this solution. Crick suggested that there must be an adapter molecule, a molecule with two functionally different ends. On the one end, he suggested there must be a mechanism for attaching a specific kind of amino acid, and at the other end there must be a mechanism for interacting with a specific sequence of nucleotide bases. And indeed, Crick was correct. There are such adapter molecules. These are small, highly specialized strings of RNA, a kind of ribonucleic acid, called transfer RNAs, or tRNAs for short. We're going to come back later and look more closely at tRNAs and how they function. But first, for now, I want to go on and talk about the second reason that our code has to be more complex than the simple substitution code like the cryptoquote code that we talked about at the beginning. And this is because there are 20 kinds of amino acids, but only 4 kinds of bases in nucleic acids. We have A, G, T, and C, and that's it. So if we had a simple substitution that was 1-for-1, we could only have a code that was specific for 4 different amino acids. But we have 20 of them to account for. What that suggests is that sequences of more than one nucleotide must be used to code for a single amino acid. By analogy, this would be like having the bases of a nucleic acid combine together to form code words where each base is a single letter in that word. So how many letters, or how many bases, I should say, must each word be for this kind of code to work? Well, imagine that we had code words that were made up of 2 letters each, that is, just 2 bases each. That would not be sufficient to code for all of the amino acids, and here's why. It's just a simple little bit of math. If we have 4 things, and we combine these 4 things in pairs, then we can make 4 times 4 unique combinations. That's 4 squared combinations, which is only 16, falling short of the 20 we need. However, as you might guess, if we make combinations of 3 things, that is a triplet code, then we would have enough unique strings, because 4 times 4 times 4, 4 cubed, is 64. More than enough. Actually, a 3-letter code might even suggest that the code would have some sort of redundancy, because we'd have a lot of code words left over after we assigned 1 to each of the 20 amino acids. Now, the logic and support for having a 3-letter code seems pretty obvious, but this logic simply suggested a testable hypothesis which then had to be demonstrated. Once again, it was Francis Crick, who we've seen many times now, along with colleagues, who demonstrated experimentally that the genetic code must involve sequences of 3 bases. Crick and his colleagues used a technique that in which they could cause a very particular kind of mutation in the DNA of a virus. This involved using a class of chemicals called acridines that would cause the elimination, the deletion of just 1 base pair from a DNA double helix, or alternatively, the addition of just 1 base pair, but only just 1, so that if they applied this treatment in the appropriate fashion, I won't go into the details, but if they applied this treatment to a virus's DNA, then they could be assured that either 1 base pair was lost or 1 was added. Now, let me explain the logic of how they used that technique in the following way. Consider a simple sentence. And you might even want to write this sentence down so that you can see it. Imagine this sentence, "Old men are fun." That sentence is composed of 4 words, each specified by 3 letters. Now, if we delete from that string of characters, for example, the first letter, and try to read it, we get rid of the O, then we've got something that spells LDM, ENA, REF, and then we've got a UN at the end, which doesn't even have 3 elements anymore. If we deleted 2 letters at the beginning, we'd still have nonsense. We'd have DME, NAR, EFU, and N. Neither of these strings make any sense. Why not? Because we have shifted the place we start reading the string by 1 or 2 positions. And the resulting remainder of the string then becomes nonsensical. But now, in this particular example, which obviously I've contrived to make the point, if we delete 3 letters, then the words come back into register and they make sense again. Men are fun. It's not the same sentence as we started with. And that's not the point. The point is that the words in that sentence, each in this case comprised of 3 letters, only come back into register and start making sense if we take out 3 letters, not 1, not 2. And we could do just the opposite. If we added a letter, even a random letter at the beginning, adding just 1 or 2 would cause the whole string to become nonsensical. But if we added 3, at least part of the string, the original part of the string, would make sense. Crick used that same kind of logic with this technique for deleting or adding just 1 base pair from a viral DNA to show that there must be a triplet code. If they induced 1 mutation in a gene from this virus, then all of the amino acids coded for that by that gene would get changed. They'd all get shifted. There would be nothing about that gene that was similar to the original gene. If they eliminated 2 base pairs, they'd have the same result. But if they deleted 3 base pairs, then they found that the remaining portion of that gene would make sense in the sense that most of the amino acid sequence would be intact, even if the one amino acid that was affected by a deletion didn't make sense. Now you can imagine this technique wasn't so specific that they could choose exactly where they could do deletions or insertions of base pairs. And so you would find a number of amino acids might be affected. But in general, if they deleted 3 or added 3 base pairs, most of the gene would make sense in the sense that the amino acid sequence would all be in register. And in doing this kind of work, it was pretty clear that the genetic code had to be made of 3 bases each. Many experiments that since that time have used a variety of techniques to verify the existence of this triplet code. And the 3 base sequences that serve as fundamental units for this code have come to be known as codons, with each codon corresponding to a unique protein. Now, the next step was to somehow establish what the correspondence is between particular codons and particular amino acids. The codon codes for a particular amino acid. Crick's work demonstrated that the code must be 3 elements long, but it couldn't establish what the relationship between any 3 elements and any particular amino acid was. So how did molecular biologists end up cracking the code? Well, the technical breakthrough that led to the deciphering of the code occurred somewhat later, in the 1960s, in the early 60s, by the development of a method for synthesizing artificial RNA polymers. In a nutshell, an enzyme had been discovered that, when put in proper conditions in a laboratory test tube, would randomly take the nucleotides that would build an RNA and, without any template, would just start polymerizing them. So you would have a set of nucleotides you put into the test tube, you'd add this particular enzyme, and what you would get out would be some random string of nucleotides that were now polymerized in an artificial RNA. Now you might ask, what use is this if you can't control the sequence? I mean, what we'd really like to do is, if we could, is we'd like to come up with specific sequences. We'd like to be able to say, give me a sequence that's CAA, 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 and let me see what that then produces when I use it as a template for protein synthesis. But back then that wasn't possible. But they still could use some clever reasoning and methods for cracking the code nonetheless. And let me show you how they began. They first began by only putting in one kind of nucleotide. In fact, the first nucleotide that they did this with was the base uracil. Remember, that's the base found in RNA that corresponds to thymine that we find in DNA. And if they put this enzyme into their preparation with nothing but the base uracil, what they would get out, of course, is one long continuous polymers of nothing but U's, a poly-U polymer. And then when they used that artificially created strand of RNA as a template for protein synthesis by other methods, they found that they would get out a string of phenylalanines. Phenylalanine is one particular kind of amino acid. Well, the only logical conclusion from that result is that the codon UUU, 3 U's, must correspond to the amino acid phenylalanine. Similarly, it was easy enough to understand how they could decode strings of A's, which actually produced strings of amino acids of lysine amino acids, strings of C's, which turned out to code for prolines, strings of G's, which produced glycines. So the first 4 codons, which were codons that had all the same bases, were easy enough to figure out how to decode. Codons that included different combinations of bases, however, were harder to decode, but not impossible. And this is where the work got to be really clever. And I'm not going to go into the details of how they did this, but suffice it to say that even though it wasn't possible at the time to synthesize a known sequence, we can do that, by the way, now very easily using the techniques we have available today. But even though it wasn't possible to do that then, if they added nucleotides of known different proportions into their reaction mixture, then they could predict what all of the possible codon sequences that would be formed would be in their own known proportions. So for example, without going into the math, if you put in a 3-to-1 mixture of A and G and allow this synthesis to occur, you predict that there would be different proportions of the possible combinations, with the largest proportion being the combination AAA. A smaller proportion of the possible codons would be AAG or AGA or GAA. An even smaller proportion would be AGG or GGA, and so forth. Now, what they would do is calculate what the predicted proportions of the combinations that they would expect to find from randomly synthesized strings was for any particular mixture of 2 or even 3 nucleotides that they would put into the mix in known proportions. Then they would allow the reaction to run. They would then use the synthetic RNA as a template for synthesizing a polypeptide, and they would ask what was the proportion in that synthetic polypeptide of the different kinds of amino acids. So this was a lot of detective work, and you can imagine that there had to be a number of these experiments done, and the probabilities didn't necessarily always match up exactly with what they got out, but by matching predicted and observed proportions of the amino acids that should be produced based on what you expected to be the relative proportions of codons produced in the string, they were able, and a lot of cross-checking, of course, they were able to use this puzzle-solving approach to infer the entire genetic code, to figure out what every combination that was possible of 3 bases in sequence would mean in terms of a particular amino acid. Now, I actually think that the decoding of this information is a remarkable milestone in the history of humankind, because it essentially gave us a dictionary really of the relationship between nucleic acids and amino acids, which is the basis on which all life depends. If you look at this dictionary, if you look at a table of the relationships between particular sets of 3 bases and amino acids that they correspond to, there are a number of things that emerge from this. First of all, as we suggested earlier, because there are 64 possible combinations, that is 64 codons, and only 20 amino acids, there's redundancy in this code. That is, some amino acids are coded for by more than one codon. For example, if you look at the amino acid serine, you see that there are 4 different combinations that all correspond to serine. Turns out to be UCU, UCC, UCA, and UCG. Note that the redundancy in this genetic code is not random, because in fact you see this for all sorts of sets. If you look at the amino acid arginine, then CGU, CGC, CGA, and CGG all are codons for that particular amino acid. So the synonyms that occur in the genetic code usually involve differences in the 3rd base in the triplet. So we have redundancy in the genetic code, but we don't have any ambiguity. In other words, each codon, each sequence of 3 bases, corresponds to only one amino acid. The codon UCU codes for serine and nothing else. And of course, this lack of ambiguity is absolutely essential if we're going to have a functional code. We have to have the codon mean a particular amino acid and nothing else. Now, I said there are 64 possible combinations, 64 possible codons. It turns out that only 61 of these 64 codons actually correspond to a particular amino acid. 3 of the possible codons are actually what we call stop codons. These stop codons don't correspond to any amino acid, but instead, when these stop codons occur in a sequence, in a sequence of genetic information, they serve as a sort of punctuation, like the period in a sentence, indicating that the message ends here. That is, signaling that the protein is complete, when you've done the previous amino acid. So we have 3 stop codons. Now there's one codon, which is an especially interesting codon. It's the codon AUG, which serves a dual function. It does code for an amino acid. It codes for the amino acid methionine. And it turns out it's the only codon that does so. So in this case, there's no redundancy. And it also functions as a start signal, another kind of punctuation mark. All messenger RNAs begin with the triplet AUG, which signals the protein synthesizing machinery to begin translating the mRNA at that point. So why is having a start codon so important? Well, the reason is that it serves as an unambiguous marker for exactly where to start reading the sentence. And thus it's a marker that defines the beginning of the sets of 3 elements that should be interpreted as codons. Now, remember back to the experiments that Francis Crick did when he was demonstrating that it had to be a triplet code. Or in fact, go back to that sentence, "Old men are fun." If you start with the O, the sentence makes sense. But let's say you had no spacing between those words, and there is no such spacing in the genetic code. You just had those 12 letters all in sequence. If you happen to start reading it, say, with the L, it would be nonsense. So for the same reason, you need to know where exactly to start defining the triplets that are going to be read as the genetic code. And this is what the start codon does, AUG. It serves as what we call the beginning of a reading frame. If every mRNA always begins with AUG, then you know that that's the first triplet, and whatever set of 3 things come after that will be the next codon in sequence. Interestingly, you might have figured this out. Having AUG as the only start codon means that all polypeptides which are synthesized begin with the amino acid methionine, because that's what it codes for. And that seems a little odd, but that's the way it works. Not all functional proteins begin with methionine at the beginning, I should say, because this first amino acid, although it's always added when the mRNA is first translated, is often removed later on, for example, by enzymes specialized to finish off the protein once it's been produced. Well, one of the most interesting things about the genetic code, I think, is that it's universal. Well, almost. I'll get to that in a second. But virtually every genetic system in all known organisms uses the exact same triplet code. The same codons are assigned to the same amino acids, and there are the same start and stop signals. The universality of this genetic code is interesting, I think, for really 2 different reasons. The first reason is the fact that because everything from bacteria to fireflies to tobacco plants to humans, because everything uses the same genetic code, we can do what we now call genetic engineering. We can take genes from one organism and have them function in another organism. The reason we can do that is that although the details of how the genetic machinery works, the processes of replication and transcription and translation, all differ a little bit from organism to organism, or may differ, I should say, from organism to organism. In all cases, this machinery recognizes the same code. So if DNA polymerase, for example, works a little bit differently in, say, a bacteria as opposed to a human cell, which it does, it nonetheless will read the same sequence in the same way. As you're probably aware, genetic engineering is commonplace nowadays, with a variety of enormously important practical impacts and potentially somewhat challenging impacts. For example, and this has been going on for years, we can take the gene from one organism and have it inserted into another organism that is able to produce a lot of that gene product. For example, we can take the gene that produces human insulin and put it into E. coli bacteria. These bacteria reproduce very rapidly, and they're able to manufacture, therefore, a lot of proteins. And if we insert the gene for insulin into bacteria, they will produce human insulin. which we can then use commercially for biomedical uses. Now, we might be able to get insulin on other ways, but this is a very cheap and efficient way to produce a lot of insulin. And this kind of genetic engineering used to produce products for biomedicine or for other commercial uses is actually quite commonplace right now. We can also modify organisms by inserting DNA so that we're changing those organisms. In the first example I gave you, we were just using those E. coli bacteria essentially as their own little chemical manufacturing plants, giving them the blueprint for producing insulin. But let's say we want to cause a particular crop plant, or to have a particular crop plant less susceptible to freezing or less susceptible to insects. We can take genes, for example, from a bacteria that produce a natural pesticide and insert them into that crop plant, and in so doing have the plant itself produce that natural pesticide, making it more resistant to insects. These are called genetically modified organisms, GMOs, which, of course, are of great importance today. We're even starting to think about modifying ourselves by adding genes from other individuals or other organisms that could help us overcome some genetic deficiency, a process known as gene therapy. Now, gene therapy is still being developed. It doesn't necessarily work well, and it's quite controversial. But here's the logic behind it. Many disease states we know have some genetic basis. The individual who expresses that disease does so, in part at least, because they have some non-functional version of a gene. If we can find a way to take a functional version of that gene and put it into the cells of that individual, then we have another source of producing the right gene product, which might substitute for the non-functional or disease-causing gene product. The details of the technology for how we do that, as you can imagine, are quite complex and are still being developed, but all of this rests on the single fact that the genetic code is a universal code. Now, there's a second, quite different reason that we should be interested in the universality of the genetic code. And that is that it brings us back to questions we started this course with about the origin of life. The shared vocabulary of the genetic code suggests that the fundamental genetic mechanisms specifying equivalences between sequence of nucleotides and sequences of amino acids arose very early in the history of life on Earth, perhaps as early as the origin of life itself. As I've said, we don't know exactly how life arose, especially how genetic systems arose, but the fact that we all share the same code is consistent with the idea that all life derived from a single, common ancestor that occurred sometime long, long ago. Now, I said the genetic code is universal. Well, almost. There are some exceptions to the code. But these exceptions are relatively minor, and in that sense, they prove the rule again. Most of these exceptions involve assigning one or two of the three stop codons in the code that we've been talking about to an amino acid, as opposed to serving as a stop codon. The exceptions also occur in cases that are likely to represent very ancient divergences. You remember we talked about the idea that those two organelles involved in energy processing in eukaryotic cells, mitochondria and chloroplasts, might have derived from some event early in the evolution of life where one cell engulfed another, with the mitochondria and the chloroplast being the descendants of the cell that was engulfed. And our understanding of this process is that evolutionary event occurred very early on. It turns out that many of the exceptions that we see to the genetic code are in the DNA that are carried by mitochondria and chloroplasts. DNA that has evolved independently on its own in the context of these organelles now residing within other cells. Okay, now that we've looked at the genetic code in some detail, we can next start to look at the nuts and bolts of how the information encoded in a gene gets turned into a protein. And we'll start there with the next lecture.